Okay, so let's go ahead and review the central dogma. The word central means it's really, really important, and dogma is kind of like a concept or philosophy. So this is like the central idea really behind biology and genetics these days. So our checklist, you need to know what is the central dogma, what is transcription, what is translation, how can I make sure to remember the difference between those two, and what are codons and anticodons. Let's get right to work. So, central dogma, you have this diagram on your note sheet. Key parts are, we start with DNA, okay? We've spent the semester talking about it. It's where we start. It's the beginning. However, the DNA gets stuck in the nucleus. In order to get it out, we're going to have to transcribe it and make RNA, okay? RNA is something that can move around the cell. It's a lot more flexible than our DNA is. So DNA, transcription, RNA. Finally, I'm going to translate it into protein. So this is the central dogma. It's on your note sheet, like I said. Go ahead and pause here a moment if necessary to finish copying it down. And you're ready, you can go on to the next question. Okay, so the next question, what is transcription? Transcription is where we're taking segments of DNA and we're copying it into RNA. So, as part of your high school career, something that's being built for you is a transcript. It's kind of a summary of the classes you've taken. It doesn't show everything you've learned, it only shows some things. Likewise, RNA is a transcript or a copy of some of our DNA. So it's just a small portion. So we can see it right here. We see it polymerase back at work, and we're going from DNA to RNA. That is transcription. Translation is different. With translation, we're changing RNA into proteins. So it's going from one macromolecule, nucleic acid, into another one, proteins. Right here you can see we're at the ribosome, so we're out in the cytoplasm, and our mRNA is changing, going through that, and we're converting it into these pieces that make up our protein. Those pieces are called amino acids. Okay, so transcription and translation, I know, they sound really similar. So you have two tricks that I know of to help you remember it. You might know of more, but here's my first trick. Transcription comes before translation in ABC order. So if you can remember that, alphabetically, transcription is first. You can remember that's DNA to RNA, and translation, which is second, comes after that. Or you can think transcripts are partial copies, just like it's partial pieces of DNA. And in translation, we're changing to a new macromolecule instead of a new language. Okay, so other words you're going to see are codons and anticodons. When we go through translation, our mRNA is copied by tRNA. So codons are what's on the mRNA, anticodons are on the tRNA. So you can see this right here. Here is our codon, our mRNA. This is a set of 3 bases. So the codon, once again, are these pieces of 3 bases on the mRNA. So this would be a codon, or sorry, GAU would be a codon, ACC would be a codon, and then GAG would be a codon. Anticodon is its partner, and it's found on the tRNA. So I hope you're looking at the diagram down below. You see the codon where it's GAG. CUC is the anticodon. Okay, so that part is what helps the tRNA, which carries the parts of the protein, match up and make our new protein. Okay, so central dogma, real quick. It's DNA to RNA, RNA to protein. Transcription gets us from DNA to RNA, while translation then takes us to protein. We talked about tricks to remember it. Feel free to look back at my tricks or make your own. Codons are on mRNA, and anticodons are on tRNA. Excuse me. So those are quick run through of the central dogma. In class we'll practice a little bit more and make sure we understand different functions between mRNA and tRNA. The next video walks you through a practice problem, and that can be really helpful to understand how to solve these. Great work, y'all!